right, good morning, everyone. Welcome to Data Wednesdays this week, one of two. I know there's another at 4 o'clock. This morning, I'm really excited to welcome Dr. Pontus Skoglund from who is a group leader at the Francis Crick Institute's Ancient Genomics Laboratory. He's originally from Sweden and obtained his PhD in evolutionary genetics from Uppsala University in 2013, and then did his postdoc with David Reich in the Harvard Medical School's Department of Genetics. His past research has focused on propelling the field of ancient DNA into the genomic era, revealing population migrations as catalyzers for the transition from hunter-gatherer lifestyles to agriculture in Europe, Africa, and Southeast Asia. He's also studied gene flow between archaic and modern humans, early human evolution in Africa, the peopling of the Americas, and sort of what he'll be talking a bit about today, the origin of domestic dogs. So I'm excited to hand it over to Pontus. Thanks.
Thank you very much, Kelsey, and the hosts for inviting me here to be join you today from Southwest London all the way to Brown. It would have been nice to come in person, of course. Hopefully that will be possible in the future. But yeah, very happy to be here and interested in any comments or questions during this talk. Yeah, so today I'd like to talk a little bit about our lab's research, recent research, in fact, on ancient canids, particularly ancient dogs, and how we can relate ancient DNA from dogs with our knowledge about human genetic history as well. So let's see. So just a brief introduction about our lab. We started about 3 years ago here at the Francis Crick Institute in London, mainly focused on ancient genomic approaches as well as population genetics. We're very interested in dogs, as I will talk about. We also have another sort of major project we're trying to do on ancient human genomes in Britain to integrate them with the UK Biobank and this really exquisite information about the genetic basis of human disease and other traits, and try to understand fine-scale evolution in the human past in more detail. Thanks to all those people. So I'll try to talk about 3 different things relating to dogs. First, a kind of comparative population history of dogs and prehistoric humans. Then I'll talk a little bit about what we know and don't know about the first dogs. And then finally, I'll talk a little bit about something we have in progress right now on wolf adaptation across 100,000 years using ancient genomes. So sort of just outlining a little bit of the rationale. Why do we use DNA to study genetic history? Well, dogs and humans alike all have 2 parents, and you can imagine if we had the sort of the grand web of everyone in the past 10,000 years, 2 parents, that would be a really useful piece of information to understand genetic history. Perhaps we could reconstruct different major sort of events of populations expanding and interconnectivity between populations as well, using such a data set. We can't get that data set exactly, but with ancient DNA, what we're trying to do, and indeed what we're trying to do with modern DNA is, in many cases, is to try to get as close to this grand web of human ancestry and grand web of dog ancestry as we can. And then looking along the genome, since we have recombination in our genomes, the ancestry of different places in our genome each trace a different path through this grander web of ancestry, our grand family tree. And so by sampling many of them, we can try to understand it more fully than if we just sampled a few of them. And so the project that's going to be the basis of most of my talk was a paper we published in October last year, where we sequenced the genomes of 27 ancient dogs, basically most of them prehistoric, adding them to about 6 already previously published ancient genomes, including from Brown's own Kelsey, the host here, contributed to some of those, which were really great to include as well. Our data set was mainly from West Eurasia, so we tried also to understand more about the questions there. But it was a great collaboration with many different groups, including other ancient DNA labs and many archaeologists that contributed to this study as well. But all the results I'm going to show were analyzed and led by Anders Bergström, who's a postdoc in my group for the past few years, who's really been leading this work. And so one of the first things we looked at was to see, well, what major ancestries or different types of dog groups can we identify in this data? And then we took those, we picked representatives of each of those ancestries, and we tried to reconstruct what we refer to as an admixture graph, which you may have heard about in the past, perhaps. Basically, that is, not just trying to reconstruct a tree describing genetic history, but also allowing for admixtures to take place. So contacts between different lineages. That turns out to be a very flexible method. It's also good to use with ancient genomes that tend to have a lot of errors in them, so it's hard to use more heavily parameterized models, and we can instead use this Admixture Graph framework to often reach quite good results. In fact, since we could boil our data down, we believe, to a few sort of like real lineages of which others are sort of derived from. 6, for example, we could search the entire possible space of up to 2 admixture events between these lineages, about 135,000 graphs. And in fact, it turned out that only one of them was an adequate fit to the empirical data. Only one of them had predicted statistics, F statistics in this case, which I can talk more about if there are questions. Predicted F statistics that were indistinguishable statistically from the empirical data. And what we saw was this really quite interesting pattern where much of dog diversification must have occurred already in the Pleistocene, that is more than 11,000 years ago. The Holocene, which is this recent warm period where climates changed really quite abruptly to be much warmer all across the world. It seems that there were already at least 5 different sort of quite differentiated, types of dogs or dog ancestries, if you want to think about it in this little bit crude way, across the world, including in the Levant, in Northern Europe, in Siberia, like the Lake Baikal area, in the Americas indeed, and also one lineage that we didn't have an ancient dog representative of, that was likely in Southeast Asia, represented by the New Guinea Singing Dog, which is a dog very similar to the dingo, which more people may be familiar with. But this suggests that already 11,000 years ago, already before there were really any other domesticates, there were multiple different dogs all across the world, suggesting that the origin must have been quite a bit into the past. And so in this graph, you can also see that other European dogs, including one from 7,000 years ago, could be best described as a mixture of a Northern European dog from Karelia here and those of the Levant 7,000 years ago. And that was really interesting to us because it might be related to the arrival of agriculture in Europe, which we know in the human DNA was driven by groups of sort of ultimately Anatolian or Near Eastern ancestry expanding into Europe, and there, of course, meeting and being connected to the local populations. But there was also a separate line of evidence that supported this conclusion that it was likely to do with the agricultural expansion, and that is when we quantified shared genetic drift using something called outgroup F3 statistics, with, on one hand, on the y-axis, a Siberian dog, and on the x-axis with a Levantine dog. All our ancient dogs from West Eurasia kind of fell along the diagonal in what we think of as an ancestry cline. Let's see. So all ancient West Eurasian dogs kind of fall in this diagonal along this contrast between shared genetic drift between these 2 different dogs from really different places. And to me, this is really sort of a hallmark sign of admixture having taken place. But we wanted to also confirm that this qualitative pattern is really supportive of such punctual admixture events. So we did some simulations. And for example, if you would only have a kind of phylogenetic structure, a tree-like structure, you would quantify genetic drift along 2 axes, you would not get this diagonal pattern. In fact, it would be more sort of V-shaped. Similarly, if you had a sort of long-standing continuous gene flow scenario, you would also not really get this pattern. You would get more of a U-shaped pattern, but only if you have a more punctual type admixture event between two different sources, and with descendant populations with different proportions of this, do you get sort of a very strict line along the diagonal. So that's really quite interesting. We think it has to do with the agricultural arrival of human groups bringing their dogs into Europe, but we're not 100% sure, but that's our current hypothesis, at least really shaping the diversity of present-day dogs. And what's really interesting is, of course, if you look at, sorry, those could be rejected, that's the only one that fits. Modern European dog breeds, many of the breeds you think of when you think of a dog, your German Shepherds, your Poodles, your Terriers, your Border Collies, all of these dogs occupy only, or sort of capture only, a very limited set of the diversity that was there in prehistoric West Eurasia. In fact, they can probably be modeled as 50-50 of approximately of these two different ancestries. So it's not so that if you go 5,000 years ago and you look at the dogs in Greece and Iberia and perhaps in Britain, where I am, that they have a different relationship to dogs today. Somehow all of that was homogenized at some point from the past until today, which is a sort of interesting process. How that might have happened, we don't understand yet. And so this is sort of an illustration approximate with what we think might have happened. On the order of more, at least 7,000 years ago, we think there was gene flow from the Near East into Europe. Most likely this was 10,000 years ago, as seen also in the human data. But at some point in the past 4,000 years or so, there was an expansion of a single ancestry across Europe to create this homogeneous nature of present-day dog breeds. And that's something that was really interesting to understand how that happened. And so this agricultural expansion is something that has been indeed, suggested based on ancient dog mitochondrial DNA, specifically with the presence of particular haplogroups associated with Neolithic dogs compared to sort of non-Neolithic dogs, putative ones. But when we look at the dogs that fall along this nuclear genomic line, and we sort of here they're colored by their mitochondrial DNA haplogroup, or sort of a nomenclature for mitochondrial DNA lineages, we don't really see a very significant association with these haplogroups, but indeed perhaps some patterns that suggest that indeed they might be markers of these expansions as well, but at the moment we don't have significant evidence of that. Maybe in the future it will be possible to see that. And I mentioned before that we're also trying to put the dog history in, sort of see it through the lens of human history. In fact, it might be cases where we might learn stuff about prehistory by using dog DNA as a sort of tracer dye, or dog ancestry as a tracer dye for human activities. And did dogs follow their human groups entirely, or did they have a slightly different genetic history? So you can imagine if we knew the history of both of them, this is sort of common way of showing population models where populations diverge and have varying population sizes and secondary contacts. If we could overlay human and dog history on top of each other, perhaps we could learn new things about their relationship. Perhaps we can even learn new things about human history specifically just by following dogs. And so a first thing we did, which is really sort of model-free, is we did a principal component analysis on most of the dogs we had. Specifically, it was the dogs for which we could find a matched ancient DNA sample of humans from the same time and place. I'll get back to that in a second. We also did our PCA based on F4 statistics for the sort of population geneticists in the room to avoid issues with ancient genomes having more errors. For example, ancient genomes can have varying degrees of errors that really influence the principal component analysis in different ways. So we did this multivariate analysis, trying to identify what is the sort of main axis of differentiation between genomes and what is the second axis of differentiation, requiring that it's orthogonal to the first. And so we get sort of this PCA of dog ancestry, but then we do the same analysis with the matched human genomes. And we take those configurations and we try to rotate them in the way that sort of minimizes the distance between the groups that are from the same time and place, using something called Procrustes analysis. And then we can put dogs and humans next to each other to see how their genetic differentiation, their structure might differ from each other. And we've sort of plotted a link between dogs and humans here also in sort of the residuals of this overlay. And we can in kind of a model-free way see, well, where do we see the biggest differences between humans and dogs? As you can see, actually, in many ways, their genetic structure mirror each other. For example, there's an east-west difference between both humans and dogs, likely reflecting some of their older structure. We can also look at the residuals, for example, which ones are the greatest, are the cases where dogs and humans have the greatest mismatch in their genetic differentiation. Interestingly, the biggest residual was observed for the steppe Bronze Age, where the dogs and the humans really don't map to the same place in this particular analysis. And what it turns out to be, we think, is that you might have heard about an expansion of steppe ancestry people into Europe about 4,500 years ago, associated with the late Neolithic or the Bronze Age. This is something that's been well documented in humans. It had a big ancestry impact, several tens of percent of the ancestry of later people. If you go back to 5,000 years ago, that ancestry would find itself on the steppe. But in dogs, while we see some type of transient such signal in an early Bronze Age dog, let's see, we don't see a sort of long-term signal of such an impact. In later dogs, there's no particular link to the steppe, to steppe dogs. Maybe this human expansion from the steppe into Europe did not sort of take their dogs along with them. Maybe there was some other difference that created this sort of more larger difference. And there were other cases of sort of disparate genetic structure between dogs and humans as well that would be interesting to follow up in other ways, even though in most cases, dog and human structures show really interesting overlaps. But you could imagine in some cases, dogs are perhaps used more as cultural or trade commodities. Perhaps sometimes, they kind of move with their own, by their own volition, they perhaps not necessarily meaning that they are fertilized and then rejoin human groups, but perhaps, dogs are quite mobile creatures, and you can imagine them crossing between different human groups without the groups necessarily being having so much sort of intermarriage or contact in that way. But another thing we wanted to look at was a particular case or a particular genetic locus that has been discussed as a main target of genetic adaptation, actually in both dogs and humans. And it's not the same locus, but both of the loci are associated with amylase, so the breakdown of starch or starch metabolism in humans and dogs. And in dogs, it's copy number of the AMY2B gene, so pancreatic amylase, which, so if you look at these different panels, in sort of a diverse set of canid species, including the African hunting dog and golden jackal, there's sort of a fixed copy number, as there is also in the absolute majority of wolves. But if you then go all the way to the right here, you can see that in present-day dogs, most dogs tend to have a much higher copy number of this variant, which, a natural explanation that's been put forth in the literature before, in references here on the slide, is that it's associated with, having a diet based off of human societies, perhaps, sort of refuse of humans or being fed by humans, starch-rich food. However, while agriculture, started around 10,000 years ago, we don't see kind of an immediate or sudden increase in copy number in the dogs we have. Even 4,000 years ago, there are dogs that are basically associated with agricultural human groups that still have a fairly low copy number. And so it was likely that this was more of a complex rise of this variant in different human groups. It probably took a while before it reached this sort of very high frequency seen in dogs today. And indeed in dogs that are still in some way associated, or that are the archaeological dogs that were associated with late hunter-gatherer groups, including in Sweden 5,000 years ago, and as well as the various American dog genomes that were previously published, they also show sort of retaining a very low copy number, suggesting that this is more of a complex process of adaptation in dogs. So I talked a little bit about a model-free approach to dog ancestry, trying to look at it and also comparing it to human, but we also wanted to model ancestry more specifically, and we turned to this Qpdm framework, which is a really fantastic framework. We can talk a little bit more about what's under the hood if there are questions. It basically uses F statistics again, and asks the question if a target population can be modeled, for example, just as being a clade with one other population to the exclusion of a bunch of others. And the advantage is that we don't have to concern ourselves with the internal relationships of all of these, indicated by this cloud here. But we can pose various hypotheses of the ancestry of the target population, perhaps also being a mixture of two different source populations to the exclusion of all others in the sort of reference or outgroup population set. And it's fine in this model if there's complex relationships between the sort of outgroups that we pose in the model. But what would be a model violation is if the target population has ancestry from sort of one of the outgroups closer in time than to the source population that's indicated in the model. And that was sort of a brief introduction, but we wanted to look at this in a wide set of present-day dogs, and we model them as having their ancestry most closely related to one of these different sets in this kind of sort of trivial pursuit pie chart here of different ancestries, including then a steppe dog, two early sort of farmer dogs, both from Iran and from the Levant. We also modeled specifically modern European breed ancestry because in the colonial era, dogs spread with European settlers across the world. And so it's kind of interesting to understand what component of worldwide dog ancestry is immediately from them, but also our other ancient dogs from Siberia and the Americas, and then the New Guinea Singing Dog, which is the best proxy we have for what we believe are these Southeast Asian dogs. You can think of it as a type of dingo, basically. And they're really, I found it really quite fascinating. So again, while we don't see a large component of steppe dog ancestry in Europe today, there's a possibility that there's a much greater proportion of steppe dog ancestry in East Asia, for example, China, which might be a different outcome than what is observed in humans, which is quite interesting to me. We also see that African dogs, such as the Basenji, and indeed with a few percent, a breed such as the Rhodesian Ridgeback, which has most of its ancestry from European breeds, but indeed a small percent of its ancestry from what we think are the sort of original African dogs, is best represented by our Levant Neolithic dog in this data set, which is really interesting to us. And actually, it's a difference from later Levantine dogs that we also have in our ancient genome data set. And so these few percent of pre-colonial ancestry, if you will, in the Rhodesian Ridgeback is also actually observed in breeds in the Americas, such as the Chihuahua and the Charoloid Squintly, both Mexican breeds, that both have sort of like 3 or 4% of, in our models, pre-Columbian dog ancestry, which is something that I've been interested in for, I was involved in a study almost 10 years ago, which had this question, is there any ancestry from pre-Columbian dogs surviving until today? And there seems to be a little bit, and other studies have also suggested this. But still there are some interesting questions about this, and perhaps also how these dogs retain their sort of large cultural importance. And indeed, many of these ancient dog sources that are in the pie chart here, did contribute in different ways to dogs today. So as I like to say, if you would go to a dog park and you see, perhaps a husky, perhaps a Pekingese or a Akita, perhaps an Afghan hound, perhaps a Basenji, perhaps an Alaskan Malamute, and a Chihuahua, those dogs all capture slightly different ancestries from dogs that lived probably 11,000 years ago. It's really quite fascinating to me. So moving on then to the second part here, which is what we know and what we may perhaps not know about the very first dogs. So archaeologically speaking, there's an interesting observation, for example, here in this figure by Laurent France et al. From 2016 in their paper, where there are the most confident skeletal representatives of early dogs pop up in sort of very different places of the world, including plausibly in Europe, some good examples, 14 or 15,000 years ago, in the Levant, 12,000 years ago, and also in Eastern Eurasia, such as the Baikal, sort of 14, 15,000 years ago, and indeed, as Kelsey is an expert on, probably with the first people in the Americas 15,000 years ago. So suddenly 15,000 years ago, dogs are all over the place. How did that happen? How could we find where dogs originated? Where were they domesticated? And how did they spread worldwide? And so there's a lot of, people have tried to look at this in the literature in many different ways. And there are a lot of very interesting hypotheses based on modern DNA. I think it's difficult with just modern DNA because this dynamic dog population history that ancient DNA is starting to reveal probably overwrote many of these things. Indeed, Kelsey was involved with this really interesting paper looking at a mitochondrial DNA lineage, which they found, and which is the most deeply coalescent one that we know of in ancient dogs at all, found specifically associated with the Americas, really suggesting that dog domestication can't have happened so much or like so far away from the Americas, which I think is really interesting as well. But we don't have sort of like a holistic view, I think, of dog origins. If we could see a pattern such as this model, where domestic dogs in gray are clearly more close related to one type of wolf today than to others, for example, here just as a hypothesis, European wolves, that would be really good evidence that maybe dogs were domesticated in Europe or at least nearby. But that's not really what we see. Instead, most models have found that dogs are an outgroup to the diversity of wolves today. And so who were these ancestral wolves that became dogs? We don't know where they lived. And furthermore, Ice Age wolves seem to also not really be associated with this lineage, the ones that we've looked at so far. They mostly seem to be an outgroup to both dogs and present-day wolves. And indeed, it looks like maybe this ancestral wolf population that became dogs was quite diverged from present-day wolves. So about 5 years ago, we analyzed this first ancient wolf genome, and we were able to use the fact that it's directly dated to 35,000 years ago to calibrate a canid mutation rate. And this mutation rate can recalibrate the divergence of dog ancestors to present-day wolves to, I think, more than 27,000 or so years ago. But it's important to note that that divergence might not be domestication, because those canids were dog ancestors, but they were very likely still wolves. So we still don't know exactly when domestication happened. So it's, I would say, between 11,000 and probably at the upper limit somewhere around 30,000 years ago. And so one of the things we did with this new ancient dog data set is to ask the question, what direction is gene flow between dogs and wolves? There's plenty of evidence for this gene flow, but what are its dynamics, what are its characteristics? And the test we set up was to ask, well, is an ancient wolf genome symmetrically related to two different dog genomes? And we used a variety of combinations then of pairs of dogs and of single wolves. And we found much evidence for deviations from symmetry. So if they are symmetrically rated, they should the confidence intervals here should fall along this dashed line. But for the red dots, you can see that it sort of significantly deviates from the null hypothesis. And so there was plenty of evidence of that for a variety of different wolves of asymmetrical relationships to present-day dogs. But we could also find some wolves that didn't show any such deviations. So no matter the pair of dogs that we asked this question about, we didn't see any deviations. And to us, that in this paper, that suggested that maybe most of the gene flow is actually from dogs into the wild, and that gene flow would not have affected wolves such as this Xinjiang wolf. If the gene flow was from wolves into dogs, we should be able to detect the asymmetry with any wolf, basically. But we don't do that, suggesting that most of the gene flow is into the wild, into wolves. Although there is a power issue here, there might be sort of subtle gene flow into dogs that we don't detect with these things, with this particular approach. And of course, I mean, perhaps a model for understanding this is that maybe wolf genetics, sort of brought with it the behavior that actually differs between dogs and wolves quite substantially. Dogs are much more easy to deal with, basically, for humans, and perhaps wolf genetics weren't so well suited to be in human societies, perhaps contributing to this pattern. But we're still sort of left with these questions. So here's a recent review of what we know about sort of Paleolithic human genetic history from 45,000 years ago until more recently in Europe. Different human groups, we know quite a bit about their history, a lot of mysteries remain, but what we can't explain with this is why do dogs pop up suddenly sort of 15,000 years ago in Europe, in the Near East, in the Americas, probably in Southeast Asia as well. We don't know of any human migration that could have facilitated this spread. And it's really a huge mystery to me, and I think also others, how this happened. We see no evidence in any of the papers so far that there were really good evidence of multiple domestications, even though that would perhaps be a natural explanation for why this happened. But all dogs today at least share some common ancestry, but maybe there are subtle signs of different domestications that we haven't picked up so far. And one of the keys to understanding this, we think, is to study wolves, because we might be able to find with ancient DNA some very early dog somewhere, but how do we know that we're not just missing an even earlier dog somewhere else that's actually the first domestication region? But if we can sort of fit in dog genetics to a geographic landscape of wolf genetics, that's what I think would perhaps be the best way to figure out where dogs were domesticated. And studying wolves would also be interesting for a second question, which I sort of put up here, in that Ice Age gray wolves, they're still around, but other in northern Eurasia, but other Ice Age megafauna such as cave lions here on the left, so this is a real specimen, cave hyenas and cave bears all went extinct in sort of very similar regions at the end of the last Ice Age. So how did wolves survive? It's kind of an interesting question to understand. And it's one of the ones we're trying to address with this kind of a new initiative where we have over 70 wolf genomes from mostly across northern Eurasia. We're trying to ask this question of, and it's again led by Anders, this analysis that I'm going to talk about, but also again with a very sort of a fantastic international collaboration network of many ancient DNA labs and many zooarchaeologists across the world. And it also includes some quite interesting specimens by our colleagues Love Dahlén and his group at the Natural History Museum in Sweden, and Dave Stanton, including this about 30,000-year-old wolf head that has this exceptional preservation in northern Siberia. It's really interesting. Let's see, do I have it also? Yeah. And actually also this another more 18,000-year-old cub, nicknamed Dogor from approximately the same site with really excellent preservation. Actually, too bad I didn't have the free, image here. And so one of the things we ask of this data set is, well, are they all consistent with being outgroups to both modern wolves and present-day dogs? A problem with asking that question is reference bias, where a lot of ancient genomes show an attraction to the reference genome we use, which happens to be a boxer dog in most cases. So here's that quantified sort of on the y-axis. So what we did in this particular analysis was to include the exact genome used in the reference and model and allow for sort of reference-specific gene flow, if you will, which we hope will account for this reference bias, which has the risk of otherwise making some ancient wolves look closer to dogs than they really are. And so accounting for this, we can sort of assign ancient genomes to either the present-day wolf lineage or as an outgroup, or indeed as present-day dogs for some of them once we knew were dogs. And as you can see, until the last glacial maximum, some 25,000 years ago, all the wolves we look at are consistent with being outgroups to the present-day diversity. But as we come out of the LGM, many wolves are consistent with being on the modern-day wolf lineage, and some we can't really place at around this time where it's still fuzzy. And so it would make sense, again, if dogs sort of arrive on the scene at about this time period, at least among the samples we looked at so far. But as you can see on the x-axis here, which is sort of the date we have for these different individuals, the oldest ones we've calibrated using their mitochondrial DNAs, the time series actually goes up to 100,000 years ago, which is a quite interesting time series to also study natural selection. In fact, it's since dogs and wolves, actually, sorry, since wolves have about a 3-year or so generation time, that's 30,000 generations. So compared to humans that have sort of a 30-year generation time, evolution is happening sort of at 10 times the pace or so. And so it offers the opportunity to sort of understand evolution through time, genetic change through time, really the definition of evolution, perhaps in a new way. And so indeed, we did this analysis where we compute a statistic that's basically just increase in allele frequency through time. And of course, all variants in the genome will have some pattern through time, but when we look at them genome-wide like this, you can see that there are clearly some variants that show some parts of the genome where many variants show a very sort of sudden change through time in this Manhattan plot that plots the p-value for the observations to be consistent with a neutral model. And the biggest peak here in this model, let's see, when we look at it more closely, you can see that so we start 100,000 years ago, and all dogs for a long time have entirely sort of one of the variants. And then there's a sort of a time between, so we only have single copies from each genome, so they can only be 0 or 1. But there's a time where both variants are present in the dog series, and then from about 20,000 years ago, we only see the other variant. So it's a pretty sudden shift of this variant and this big peak that also affects linked nearby variants, as you can see in the Manhattan plot. And it's quite interesting locus. I mean, there's a lot of storytelling you can do about these types of analysis, but actually this gene where the signal is IFT88 is involved in craniofacial development in mice, and actually a candidate gene for human cleft lips and palate, suggesting that it has something to do with craniofacial development. And indeed, in the fossil record of wolves, people have noted that perhaps past ecomorphs were more adapted to preying on megafauna. And so basically they were bigger, have sort of bigger jaws, bigger teeth, but maybe that changed perhaps at around this time, and suddenly they had to adapt to smaller species or had a different sort of adaptation optimum. That's at least a story I would tell about this variant. But I think it's a pretty exciting approach that I hope in the future will also be applicable to other megafaunal species where we can get a lot of ancient DNA. We actually have more ancient Pleistocene wolf genomes in this study than in the total published human literature. So that's a quite nice thing that there's a big fossil record of wolves, at least from Siberia, that we can use for this. So with that, that was what I wanted to talk about, and I'm happy for further discussions and questions if there are. Thank you.
Thank you very much. So we have some time for questions. I actually wanted to start with a question I had, if that's all right, Pontus.
Of course.
Can you actually just go back to the previous slide?
Yeah.
I was curious if the skeletal material you got these genomes from are complete enough that you could actually look at the skull and look at the skulls and compare if there is a phenotypic difference for those genotypes. Is that something that you've been able to see?
I think that would be super cool. I think it's not complete enough, unfortunately, but maybe it doesn't have to be exactly the same individuals, and maybe one could have a second look at the fossil record as a whole and see, you know, like this shift is dramatic on the, I mean, it's a large evolutionary timescale, so it's actually, maybe it's actually not that dramatic, but since we look at it at the scale, it looks dramatic. And so you should be able to see it. I agree that that would be really cool if there are specific features you could find.
Yeah. Does anyone else have questions?
I have a question, Emilia, here about this slide. So you said that you were looking for changes in allele frequency. Maybe I misunderstood. Do you have multiple samples from like the same time period, or?
Yeah, no, yeah, it's a little bit trying to get around some of the problems. Great question with ancient DNA. And so basically we're just using this data here, but not the modern ones that are less than 0 here. So we're using this time series here, they're either 0 or 1. And we basically just ask, is there a correlation between this allelic state, 0 or 1, and time? And then we compute the p-value for that, and that's what you see up here. It's a very crude method. Actually, it would be nicer to actually have some more deterministic model of natural selection, like having this kind of sigmoid shape that you would expect. But for now, we're doing this kind of quick and dirty approach. Yeah.
I see. So it's presence or absence over time, I guess.
Yes.
Okay. I see. I guess I was wondering maybe that if that's the case, then could that be maybe that could be a conserved region, right? That's conserved through time and then it shifts.
So yeah, it could be conserved before perhaps, and then yeah, some type of shift. Yeah, I mean, so genetic drift, if you just look at this panel, right? Genetic drift could certainly result in these patterns. But we don't see that for any other variant, really, you know, a strong signal in the genome. And indeed, it's like a lot of linked variants to this one that seem to show the same pattern. And that's why we think it's an outlier, at least. Yeah.
Okay. I had another question too about what you said earlier in your talk, that around 15,000 years ago, dogs appear everywhere. So is there like an ecological reason for that? Have you thought about or why did they have predators that disappeared? I don't know. Just any thoughts that you may have on.
Yeah, I mean, well, it is after the end of the last ice age, but it was, I mean, it's still not as warm as in the Holocene. So like, I mean, I would say that most human societies hadn't really shifted, you know, like their ecological niche that much. I mean, in some places of the world, for example, the Near East, that would later be an agricultural center at 10,000 years ago, people were already starting to manipulate their environment a little bit in what archaeologists call the Epipaleolithic. They would sort of like come back to the same spot and sort of harvest the seeds there, and like it would sort of be like semi-agriculture, but not entirely controlled. And so you could imagine that those people, you know, they could have been in other places of the world. You know, maybe it had something to do with them being more involved in the environment. I don't know, or maybe indeed, maybe the wolves got, I don't know, friendlier or smaller or something that made it, you know, easier to deal with. I don't know. That's interesting, I guess. Well,
I have a question. Great talk, Pontus. I really enjoyed this. As a dog owner, I'm quite fascinated with this. On this slide that's up, I'm curious about the sort of an epistasis question or an integration question. Are the other peaks in any way associated with the so-called pathway that may be associated with craniofacial development? That is, if you used a network approach, can you pull in these other peaks, or are they totally independent functionally from any kind of annotation? I mean, you don't know what the gene is because it's this is a windowing approach, right?
This is actually single SNP statistics you're looking at. We
Okay.
don't, but still, yes. Yeah, we haven't gotten very far at this, basically, is my answer. Yeah, that's very interesting. Yeah.
And
I think we have a list of all of these outliers, and we haven't done a proper network analysis. I mean, it would more be sort of like if I don't think there's anything like as clearly associated with craniofacial development, sort of just looking at it and sort of Googling what the genes are involved with in the scientific literature.
Yeah. And on a related question, sort of my own pet question is, for the patterns that were derived from the earlier mitochondrial genomes and now your new nuclear genomes, is there any hints of association, functional associations between those two markers, or is this is the discord that you implied one we would expect from just two randomly unlinked loci? My question really is about whether this functional association between mitochondrial and nuclear loci in dog, metabolism, function, fitness, and so on.
I'm not aware that it is, and I mean, but on the other hand, it doesn't exclude it really. I mean, dogs are, the second species to sort of be important in direct to consumer genetics. So there's an increasing sort of medical genetic literature and genetic basis of traits in dogs. I'm not aware that it has been linked, but I
mean, if it were strong, it would have jumped out, but other people would have found it. Yeah, right.
Yeah.
Great, wonderful talk. Thanks.
Thank you.
I had a small question. I was going back to the observation that there was very little gene flow observed going from wolves into dogs. I was wondering, how much of that might be explained by their relative population sizes. I guess I don't have a good sense of what those population sizes are, but I was curious if it was lower, even given the expectation with that.
Exactly. So yes, I completely agree with you. Maybe the dogs just sort of like overwhelmed in numbers the wolves. That's possible. I don't know if the archaeological record supports that. Also, so the effective population size of dogs is actually much smaller, probably because of the domestication bottleneck. So that's a little bit of a problem for this analysis that I show, because that means that it's easier. So dog genetics is more distinguishable because it went through this bottleneck. So we can find it in the wolves easier than we can find wolf ancestry in dogs. We have less power for that. So yeah, it's two ways. Yeah, it's a great point.
Yeah, thank you. That's a great talk.
Hey, Pontus. I had a question. I found your results on amylase in just the progression of that, the increase in copy number, very interesting because it matches some of the results in Ian Matheson's papers too, how selection for some of these genes that were expected to rise in frequency very immediately after farming, but it seems to be some kind of delay. And I was wondering if you had some thoughts as to why there seems to be this kind of delay both in humans and in dogs.
Yeah, exactly. Well, yeah, maybe amylase in humans is a little bit complicated as well. Lactase definitely in humans, right? You would maybe you would have naively expected lactase persistence to rise very rapidly in humans with the onset of agriculture, but exactly, that seems to be much later as well. I don't know. Yeah, maybe it takes a while, for these adaptations. Maybe we can't really expect such a sudden shift overall with the effective population size in humans. Maybe that's just how it is. Or maybe the transition is more gradual for these things. I agree. Yeah, I don't know what to say. It would be interesting to. I don't know if you have any thoughts.
No, it's a very interesting problem. I don't know if it has to do with biases in the archaeological record. So maybe, I don't know, if I have no idea if this is based on like maybe pollen and, maybe the amount that is actually being farmed is smaller. But it's smaller early on, but there's some bias because pollen is easy to find or something like that. But I have no idea.
I mean, in the dog case, it might also be that there are different dog populations, and some of them are not really associated with agricultural populations at all. But in humans, yeah, I mean, I guess by sort of a few thousand years ago, most people had a quite starch-rich diet. So yeah. Yeah, thanks.
Hi, Pontus. Here, Maria, I have a question. Well, great talk. First, I wanted to ask if there's anything you found in the MHC in dogs that is worth like noticing. Have you seen anything like since you found these parallelisms between humans and dogs, and we've seen some changes in history in the MHC? I wonder if you've seen something like that in dogs.
Hi, Maria. Yeah, that would be. We haven't even looked at it. Yeah, we're very in the beginning phases of looking at selection in dogs. That would be, and an MHC is sort of famously difficult to deal with, so that only experts like many people in this room are able to deal with it. So we have to, I mean, that would be cool. I actually don't even know the modern literature about MHC variation in dogs. I must admit, that would be really interesting as well. Again, I'm quite excited about the dog, studying dogs, because there are more and more genome-wide association studies in dogs, and there's much functional understanding of the dog genome that maybe, you know, it can be a quite cool model organism in the future.
Yeah, I agree. Thank you, Pontus. Great work.
Thanks.
I had a question about the sudden emergence of dogs. It's just really speculative, and I don't know if the human data would be there, but does it correlate in any way with changes in human behavior, such as hunting tools or different methods of hunting that they might have used dogs for?
Yeah, I guess my answer might be that, yeah, maybe if we actually figure out where the origin was in more detail, maybe we'll see that there are, yeah, exactly. You could imagine that dogs is a new technology and there are other technologies that go with it in a way. But yeah, I don't know, maybe other people are aware. Yeah, sorry, I don't have a good answer, but that's a great hypothesis actually.
All right. Well, thank you, everyone. Thank you, Pontus, for sharing your research. I think it's really, really cool stuff. Oh, sorry. Go ahead, Amelia.
Do you have a dog?
No, I mean, I grew up with a golden retriever, but yeah, I don't have a dog recently. I'm probably more of a cat person, but yeah, they originated in very southern latitudes. It's hard to study them.
Thank you. Great talk.
Thanks.
All right. Any other questions? All right. Well, thanks for joining us this morning, and thanks again, Pontus, for sharing your work. It's really cool stuff, and I'm glad you could come and be with us virtually today.
Yeah, thanks all, and thanks to meet new people and see many familiar faces. Really exciting in these times. Thank you.
Thanks. Have a nice day, everybody.
Bye.
Bye.